Yeah. Okay, so this portion of the presentation is on immunohematology. So I'm Sherry Dunn. I'm faculty at the Department of Medical Technology, and we're also known as medical laboratory scientists. So when you watch TV series like House, and you see the doctors performing cross matches, well, that's just not done. It's so unsafe to have any other personnel in the hospital perform a cross match other than those that are highly trained to do it. So in immunohematology, or blood banking as it's more commonly known, it's the only portion of the laboratory where we're giving something to the patient instead of taking things out of the patient. So we're taking a donor unit and transfusing it into a recipient. So let's get started. But before I can talk about blood transfusions, I have to remind you that in the early days of medicine, bloodletting was commonly practiced, and that was considered normal. And this goes way back. You can even see petroglyphs or hieroglyphs in Egypt depicting people getting lanced to bleed. Okay, so that was their idea of healthy practice, was to go and get blood. And it's as routine as getting a haircut, because guess who has the sharpest tools? The barber would have the sharpest blades, right? So can anyone see the relationship between these images? What's here is called a lancet. You know what that stands for? The notches to put your arm, and then the blood would collect into that bowl. And what's this? You know what that is, right? It's a barber pole. So imagine the early days of bloodletting. You go to the barber, get your hair cut, and while you're there, you get lanced and you get bled. Well, the barber would have to wrap your arm with bandages. The bandages would get bloody. Those bandages were hung from a pole in front of the tent or shop or whatever. And that was advertisement to let people know they're open for business. So that's the origin of the barber pole. So these are your bloody bandages hanging from the pole, letting people know you can come for a haircut and you can get lanced. So people thought that there were 4 humors, and blood is one of those humors. And since it's easy to get bled, that's how you balance your humors. So for the longest time, that was very normal practice, was to go and get bled routinely. So how many of you know the story about George Washington? Anybody familiar with that story? No? He had a really awful sore throat. It's assumed that he had strep throat. It was obstructing his windpipe. He had trouble breathing. And what was the first thing the physician wanted to do? He bled poor George. He didn't get better. His fever was raging. He still had trouble breathing. So they asked for another physician to come. The second physician says, you didn't bleed him enough. So proceeds to lance George Washington again, bled him some more, and I think he lost more than half of his total blood volume. And so the poor man exsanguinated to death. Who knows if he would have recovered from his strep throat, but certainly losing blood hastened his death. So bloodletting was practiced for a long time, and it wasn't until the medical renaissance in the 1700s when somebody named William Harvey was able to study the blood vessels and valves of the human body. For a long time, the church prohibited people from studying cadavers. They thought it was defiling the body. And so until the church relaxed their taboo looking and dissecting cadavers, we really didn't know that much about circulation. And so William Harvey dissected, studied the blood vessels and valves, and he found that if you put fluid into these veins and arteries, it only moves in one direction. It's because of valves. The valves prevent blood from flowing back. Jean-Baptiste Denis is mentioned here because actually he's the first person to reportedly transfuse blood. But it was kind of a bizarre experiment. It was this lamb's blood to human transfusion. Not something I would recommend anybody to try, because there are all sorts of antigens that are so foreign to a human being that would cause immediate anaphylactic shock or death. So imagine, since the days of the Egyptians, they were leaving people, and now all of a sudden they're thinking, oh, maybe this will be a new way to treat people. Because Jean-Baptiste Denis was a physician to this young man who was going crazy. He was running through the streets naked and screaming. He was pretty violent, and he thought maybe if we transfuse lamb's blood, the characteristics of the lamb will be transmitted to the recipient. Well, that young man got very sick. He almost died. And after going through some pretty traumatic recovery, he was better. For some reason, maybe that fever was able to kill off some of the bacteria that was causing his dementia. So we don't know that. But the young man started to experience very dark urine. And later on in this presentation, I'll explain to you what that's all about. So he was reacting to that transfused blood, not in a good way, but we can credit Jean-Baptiste Denis as being the first physician to record a transfusion. So here's that lamb, and they transfuse into that young man. This is Harvey dissecting blood vessels and examining valves of blood vessels. So it was more common to kill people transfusing blood, whether it was from an animal or from human to human. And nobody really understood why. Yet, every once in a while, there'd be success. A human to human transfusion where the recipient seems to recover. But more than likely, a human to human transfusion was going to cause more trouble than that. So there are a lot of challenges to transfusions. When you remove blood from a person, it wants to clot. And then once it's removed, it can't be preserved. People really didn't understand what blood was or what it needed in order to prevent it from clotting or to be preserved. Transfusion devices were very complicated. And as I mentioned, there were severe reactions by the recipients. It was more common for the recipient to not survive the transfusion. So here's an image of one of those transfusion devices. You'd have to have the recipient and the donor close to each other, and then the cannulas are inserted into the veins, and an assistant would work the syringe, drawing the blood from the donor and then pushing the transfused blood into the recipient. So it was kind of a two-man tag team. So for years it was hit or miss. Nobody knew when transfusion would be successful and it would be fatal. And it wasn't until the early 1900s when Dr. Carl Landsteiner and his colleagues studied the reactions between red cells and plasma. So what he found through a series of mixing studies, there's only two determinants of blood groups. You're either A or B, or you lack A or B. So because of his studies, he was able to categorize blood groups into A, B, and O. And it wasn't until about 1904 when a colleague found AB, where a person's red cells possess both A and B. Okay, so here are all your blood groups discovered and categorized by Dr. Carl Landsteiner in 1909. A, B, AB, or O. Those are the four blood groups. So he won the Nobel Prize for that. And what he did was he studied hemagglutination. So hemagglutination, it tells you exactly what it is. Heme being blood, agglutination is clumping. So by the mixing experiments, he discovered some patients' red cells will agglutinate in the presence of dissimilar person's plasma. And I'll show you some of those examples later. But all of blood banking, whatever we're doing in blood bank, is based on hemagglutination. We have lots of different tests, but they're all using the same principle of red cells reacting with an anti-sera or a person's plasma. And we look for those cells clumping. And here's a photo of test tubes, and we hold them over an agglutination viewer, which is a mirror, a concave mirror, so it kind of magnifies the image, and we look to see that there's clumping, and we can grade the clumping. So what was it that Dr. Landsteiner and his colleagues were looking at? Well, they categorized the blood, and they have the A, B, O, and AB groups, and then they have anti-A and anti-B. So they took the plasma from patients that are B and they possess anti-A. They took the plasma from A patients that are anti-B. Okay, so we consider these reagents because we know that it's anti-A and anti-B. And in the presence of anti-A, A red cells will agglutinate. If you mix A red cells with anti-B, they do not agglutinate. Okay, so if you have a B red cell, you mix it with anti-A, no reaction. You mix it with anti-B, it agglutinates. O, which possesses neither the A or B antigen, will not agglutinate in anti-A, will not agglutinate in anti-B. AB individuals inherited both A and B antigens, so the red cells have both. You mix it with anti-A, it agglutinates. You mix it with anti-B, it agglutinates. So that's pretty much it in a nutshell, how we do forward typing. And these are the tools of the trade. The anti-sera that's manufactured nowadays, the anti-A is blue, and the anti-B is yellow. It's to help the tech organize their work area. We use glass tubes, and then a centrifuge, which is a centrifuge that's calibrated to look for hemagglutination. So we mix our patient's red cells with the anti-sera, and then we place it into the centrifuge. where it's spun for whatever time is specified on the instrument. And when we take it out of the centrifuge, see that the red cells have settled into a button. So the cell button is along the tube bottom. You gently release that cell button from the bottom by gentle agitation. And then you look at the reaction. So the blue, remember I told you, is anti-A, and the yellow is anti-B antiserum. There's this huge clump of red cells. There's no clumping in the anti-B. So this is a blood. Okay, so remember, anti-A will agglutinate A red cells, anti-B will agglutinate B red cells. So strong agglutination with the anti-A, no agglutination with the anti-B. This has to be A. Got it? Okay, let's see if we can identify the foreign cells that will show you next slide. So forward typing is when you add known typing antiserum to cells of unknown antigen type. Let's read these reactions. It's a strong agglutination with the anti-B, no agglutination with the anti-B. What type is this? A. No agglutination with anti-A, strong agglutination with the anti-B. B. Oh, no agglutination with either antiserum. O. Agglutination with both anti-A and anti-B. AB. We've identified the 4 ABO types that Carl Landsteiner and his colleagues identified. Simple. They're all with exceptions to the rule book. That's for medtech students to discover. Okay, so our ABO system, in addition to looking for the antigen on the red cells, Carl Landsteiner and his colleagues also discovered everyone is producing naturally occurring antibodies to the ABO system antigens. For some reason, we're exposed to whatever it is in the environment, bacteria, pollen, plants, whatever. Those antigens that are similar to the ABO antigens is causing people to make antibodies naturally to the antigens that they do not possess. How many here took immunology or are taking it? Okay. Immunology, if I boil it down to its simplest form, is the person's immune system must recognize what is foreign. You would not normally make antibody to antigens you possess. You make antibodies against things that you lack. Okay, so remember that. You make antibodies to antigens that you lack. Okay, so I mentioned that people are exposed to whatever it is in the environment that elicits our production of antibodies to the A or B that we do not possess. The trouble with these antibodies is that it binds complement, and if you know anything about complement, it's the immune mechanism to destroy foreign matter, foreign bacteria, foreign viruses. Our body has an immune system that's designed to search, tag, and destroy. So if complement finds a foreign red cell in your body, it'll attach to it, and at 37 degrees, it's lysed. The membrane is destroyed. Your body just wants to take care of foreign microorganisms that way. Imagine if you have a bacteria and your body recognizes that it's foreign, and the complement system is also involved in that immune system. It's going to try to destroy that cell. Okay, so remember the story about that young man that received max blood and he started having very dark hair? It's probably because of hemolysis. Now there was an experiment with chicks that were raised in a sterile environment. These chicks do not make the antibodies to A or B because they were not exposed to the normal environment where they are exposed to bacteria or pollen. So this plasma from people can be used to sort of verify what our forward AB typing is. Because it's predictable, it confirms in the lab, and we can use the patient's serum and red cells of known antigen group to do this. So reverse typing, here's a chart that's illustrating that. If I have the recipient's forward blood group in the first column, here's what's in their plasma. So A individuals make anti-B. They lack the B antigen, so of course they want to produce antibodies to antigens that they lack. So if I add this patient's plasma, which has the anti-B, and I added it to reagent A red cells, there's no agglutination. And if I add this A individual's plasma to B red cells, there's a strong agglutination. Okay, so my A individual makes plasma that contains anti-B antibodies that will agglutinate B red cells. Okay, we understand that row. Let's look at the next row. If I'm a B individual, I would make anti-A because my red cells don't have A antigen. It's foreign to me. So my plasma, which contains anti-A antibody, will agglutinate A red cells. But if I mix my plasma with B red cells will not agglutinate. O individuals, what's on the red cells? Do they have A? Do they have B? No. They lack both A and B. So what would you expect to find in their plasma? Both anti-A and anti-B. So if I take an O individual's plasma and I mix it with the A red cells, there's agglutination. If I mix the O individual's plasma with B cells, agglutination again. Okay, AB individuals. They're the ones that possess both A and B antigen on the red cells. What would you expect in their plasma? No antibody. Because why should I make antibody to something that I possess? A or B antigen, right? You don't do that. So if I mix the AB individual's plasma with my A red cells and my B red cells, no reaction. Okay, so look at this. It's an antithesis to what we did in the forward grouping. It's confirming to the individual performing the forward typing that, yes, this is the A individual because I can detect the anti-B reaction and lack of reaction with the B cells. Okay, are you thoroughly confused? Or is there something I need to clarify here? No response, so I don't know what that means.
That means we got it.
Okay, we got it. So let's look at the tools of the trade. And here is the reagent A cell and the reagent B cell. Okay, so this is your red cells of known antigen composition. Again, we use test tubes. We place the reagent red cells in the tubes, and then we add our patient or recipient's plasma, which we don't know what kind of antibody they have. We add the patient's plasma into the tubes, put it into the centrifuge, let it spin, and when we take it out, we see again that there's a cell button. Okay, so gently release that button from the bottom of the tube and you look to see if there's agglutination. No agglutination with A. Looks like there's agglutination with the B cells. Okay, so do you remember who makes anti-B? A individuals make anti-B. Okay, so let's see if we can identify the groups. No agglutination with A, agglutination with B. A. This person's an A. A individuals make anti-B, so our B red cells are going to agglutinate. Agglutination with A red cells, no agglutination with B red cells. Who makes anti-A? B individuals. There's agglutination with A, and it looks like there's a weak agglutination with B. So if both A and B cells agglutinate, it's an O individual. The O person makes both anti-A and anti-B. Right? Because their red cells lack the A and B antigens, so they're going to make antibodies to both. No agglutination with either the A cells or the B cells. What kind of? Okay, it's an AB individual. AB individuals have A and B antigens, so they're not going to make antibodies to A or B. Yes?
Can you repeat how you actually did this again? Because you went just like my fast.
Which one? The previous one?
The first one you said, a little bit back.
This one?
A little bit back. And this one from here, so we're adding red blood cells to plasma?
Your patient's plasma is added to reagent red cells.
Okay.
Your reagent red cells, you know that it's an A. You know that it's a B. You don't know what kind of antibodies are in your recipient. Not yet.
So.
Okay? And as I mentioned, you're using the reverse typing, which is adding your patient's plasma to reagent red cells, as a way to confirm the forward typing. Okay, so if I know what my forward typing is, I better darn well see an appropriate reaction going the other way. If not, then it's telling me that we either have a problem or we need to search for a subgroup. I'm sorry.
Is that a standard procedure?
Yes. Anytime you have ABO typing, you're doing both a forward and a reverse. You're looking for red cell antigens and you're looking for plasma antibodies. Okay, so don't get mixed up now. Antigens are on the red cell, antibodies are in your plasma. Okay, don't get those two terms mixed up. And I'm sure it's going to be on there. So know your antigens and know your antibodies. Okay, so naturally occurring ABO antibodies. That is the reason why we had all those failures in the early days of transfusion. Remember I said complement likes to attach itself to foreign microorganisms or foreign proteins or foreign viruses. Complement also is involved in lysis, which is cell analysis. And it does this really well at 37 degrees. So when you have all these red cells lysed, it can damage your kidneys. So a lot of patients that receive ABO incompatible blood, unless there's a lot of intervention, they probably go into kidney failure. And of course, cardiac function is impaired because inside the red cell is a lot of potassium. And when those red cells lyse, it releases potassium, and potassium is not good for the heart. It can cause arrhythmia. So here's an example of hemolysis. So in the first tube is a normal serum. It's yellow, kind of clear. And then each tube to the right is progressively more hemolyzed. So as the red cells burst, they release more and more hemoglobin, and it colors the serum. In cases where we suspect a transfusion reaction, we'll collect urine and we'll spin it down. Because there is a difference between hematuria from, let's say, a urinary tract infection versus a true hemolysis. So here's uncentrifuged urine, and we centrifuged it, and here's the first centrifuge tube has intact red cells that centrifuge to the bottom. Whereas in this one, which contains hemolyzed hemolysis, you spin it down and it's still red. It's because, you know, all those red cells have ruptured in vivo. Okay, so this indicates to me that there is a urinary bleed, and supernate looks clear, so I'm not worried about a transfusion reaction. This, however, alarms me because it tells me there's hemolysis in the blood vessels, which is bad news for the patient. Okay? Well, now that Carl Landsteiner has determined the different ABO groups, and people realize, well, we better make sure donor and recipient match before we transfuse them. Successful transfusions are happening all the time, except in certain cases. There were always a few things that happened that concerned the physician. Because despite transfusing ABO-compatible donor blood with a recipient, there were still some hemolytic reactions. And so the physicians are wondering, is there another group that we didn't identify? So more studies were searching for different groups. And the doctors discovered that in a lot of these cases where there's female patients that were pregnant and they had fetal demise, that's where the fetus does not carry to term. And when they examined the fetus, it was very edematous, very swollen. They called it hemolytic disease of the newborn. And Dr. Landsteiner and Wiener teamed up to study this in the late 30s. In an experiment, they got rhesus blood, rhesus monkey blood, injected it into guinea pigs and rabbits, and then they collected the serum from the guinea pigs and rabbits. They used that serum on a random population, and they tested the blood. They found that it agglutinated 85%, and 15% did not agglutinate. They called this the rhesus factor. Rhesus factor. Well, this is fetal demise. You see how swollen that fetus is. And rhesus factor was taking blood from the rhesus monkey and injecting it into the guinea pigs. RH disease, there's mild forms of that. And if you can put the baby under UV light, you can break down the bilirubin, which is a byproduct of cell hemolysis, and then the baby won't get what's called kernicterus. Kernicterus is when there's so much bilirubin, it starts to attack the brain, and then the baby will suffer neurological damage. So to prevent that, they put the baby on the bilirubin lights. Okay, so anybody recognize this individual? Monk, studied sweet peas, Mendel, yeah, Gregor Mendel. So in 1865, Gregor Mendel's publication on sweet peas was the first time that anyone described inheritance. and expression of traits. You can use what he did for sweet peas in the study of EBO groups, because all the antigens on your red cells are inherited. You can thank your mom and dad for that. The genes are a unit of inheritance, but the variation is due to alleles. You inherit one allele from your mom and the other allele from your dad, and depending on the combination of the two, that will determine the expression of your red cell. So, okay, two alleles. Remember that. And because blood group genes are codominant, you're going to see expression of A and B in those individuals that inherit both A and B. The phenotype is what you can detect. I can test the red cell and I can phenotype it, but I have to guess what the genotype is. The genotype is a genetic composition, and I'll explain a little bit more about that here. If I'm an A individual, I can test it in the lab and I can say, yeah, this person forward types as an A. But I really don't know if this individual received A alleles from both mom and dad. Or maybe mom gave the A allele and the dad gave the O allele. In this case, it expresses as an A. I really can't tell the difference between the two. It tests as an A. This B individual, same thing. They may have inherited a B allele from both mom and dad, or maybe one parent gave a B and the other gave a O. And in both cases, the phenotype's the same. Can't tell the difference. But the AB individual has both A and B antigens. So what is the genotype? Has to be AB. The O individual has to be OO. So in this case, we know the genotype is the same as the phenotype, and there's just no exceptions to that. So Punnett squares. I'm sure in biology we've done Punnett squares. Let's look at a parent AO and a parent AO. What's the offspring's ABO group? Well, here's the probability. We have AA, AO, AO, and OO. So 1 out of 4 children from a mating of AO parents can occur. And the 3 siblings here would phenotype as an A, but I wouldn't know the difference between the AA versus the AO individuals. Okay? But what if we made an AA individual and an AO individual? All the offspring would have A expression. And I can't tell the difference between the homozygous versus the heterozygous. That's another term you may want to look up. Homozygous means you have double expression of the same allele. Heterozygous means you have different alleles. In the United States, we have a distribution that suggests O's are the most prevalent. Okay, so this is nationwide. O's about 45%. And the next group is about 49% A's. Then B's are 10% and AB's are 4%. Now, ABO is proportionate according to geographic distribution. If you look at Asia, Asia has a lot more A's and B's. And so the proportion is going to look different than that. So what if we look at Hawaii's distribution? because we're kind of crossroads to the Pacific. We have a lot of Asians here in Hawaii. You notice that the proportions are no different. So it's almost the same, yeah? O's and A's. And the B's are much higher in Hawaii because of our Asian population. So 17% versus 10%. So here's nationwide, here's Hawaii. So how many here know what blood group they are? Do? How many of you are A's? Oh yeah. You get a lot of phone calls from Blood Bank of Hawaii. Yeah? A's and O's get phone calls a lot. Now in the early days of blood transfusions, all that was transfused was whole blood. You collect blood from a donor that contains red cells, white cells, platelets, and plasma. And then that whole blood unit was transfused into the recipient. But, you know, really, why should you give all of that to an individual that only needs red cells, or an individual that only needs plasma, or an individual that only needs platelets? In the early 30s, Dr. Cohn discovered that you can separate the whole blood into red cells, plasma, and platelets. And then you can store them at appropriate temperatures to extend the life of the products. Here in Hawaii, we have Blood Bank of Hawaii, which is our only donor center. On the mainland, it's Red Cross. You probably heard of Red Cross. But here, Blood Bank of Hawaii services all of the hospitals in the state of Hawaii and in the Pacific region. Only about 2% of Hawaii donates. That's pretty small. You know, when you look at other states, their proportion of donors to population is much higher. And I suspect some of that may be due to the Asian population, where a lot of women don't qualify to donate. You have to be 110 pounds in order to donate. Packed red cells have a maximum shelf life of 42 days. And so you really have to watch your inventory and turn over those units. Platelets have the shortest shelf life, only 5 days. So if you're 18 years and older, you weigh 110 pounds or more, you can donate. Anybody here 110 pounds? No? Okay. What's your name? Okay. Now you stand up, show people how big you are. 110 pounds. She contains 10 units of whole blood. 10 units. Okay, each unit is about 450 to 500 milliliters. So, coursing through her body, she has about 5 liters of whole blood. Can you imagine taking a unit out, or taking more than 1 unit out of an individual her size? The donor will not feel very good. Okay, so that's why the donor requirements state you have to be 110 pounds or more, because that 450 mils of blood removed from you at one time, it can affect you. If you received a tattoo or piercing recently, you cannot donate. You have to wait a whole year. You know why we ask people to wait? What do you do when you get tattoos or piercings? You get stuck with a needle, right? You get stuck with a piercing. You get stuck by a needle millions of times when you get inked. So in order to avoid getting an infectious disease like hepatitis or HIV, we want to make sure that the donor is clear of that. So if you recently got a tattoo or piercing, you may not feel like, oh, I'm healthy, I'm fine, I can donate. Well, are you? It might take months before you start to have symptoms. So you need to wait a whole year. Also, if you travel to parts of the world where there's malaria, you have to wait a whole year before you can donate it. So that's places like the Philippines, New Guinea, Southeast Asia, parts of Africa. You have to wait a whole year. Well, if you meet those requirements, you're asked to sit down with an interviewer and you have to answer many very sensitive questions. And it's to help identify any health risks. And after going through all of those questions and you answer it to the satisfaction of the interviewer, then you get a physical, which is the blood pressure, pulse, temperature, and hemoglobin. We want to make sure that there's safety for the donor as well as safety for the recipient. That's why all of these things are important. So if you qualify to donate, you can either donate whole blood, which takes about 30 minutes tops. They don't want to take longer than 30 minutes to collect the blood from your arm because there's that risk of clotting. Or if you qualify to donate platelets, it can be placed on a platelet-pheresis machine. So it removes whole blood from your arm. It goes through this centrifuge that will remove just the platelets and send everything back to the donor. So you're not losing red cells, you're not losing plasma. You're just donating platelets and a little bit of plasma. This takes about an hour and a half on average to donate platelets. Some people with really good high platelet counts might take an hour. So anytime you donate, a portion of that donor's blood is sent to get tested for infectious disease. And then the whole blood is separated into red cells, plasma, and platelets. Plasma must be stored frozen in order to maintain the viability of the coagulation factors. So plasma is a great source for all those factors to help with clotting. Platelets, also involved in clotting, must be kept at room temperature. And the red cells must be refrigerated. So here's a blood collection bag, and there's a main bag and then there's satellite bags. And we'll see why we need satellite bags in just. So the donor blood gets sent to the component processing area. The tech will examine to make sure that everything's labeled properly and show weigh it. Then place it into a centrifuge. And it's a light centrifugal spin. It's done at a lower speed because we want platelet-rich plasma. So here, after it's centrifuged, she takes it out of the cup, and you can see that the plasma is on the top, and the red cells, because they're heavier, are on the bottom. So this is the platelet-rich plasma that is separated into the first satellite bag. So these devices are just spring-loaded clamps, I guess you call it. The bag is placed in that, and a spring will close the two boards together and will force the plasma up the top of the tube and into the satellite bag. Okay, so now that the satellite bag contains the platelet-rich plasma, the tech can separate the red cells from it. So using a heat sealer, she will heat seal the line between the satellite bag and the red cells, and these are ready to be refrigerated. The platelet-rich plasma is placed into the centrifuge again, and this time it gets a hard spin. It's spun at a much faster speed. And then all of the platelets will be packed in the bottom of that bag. Once again, the bag is placed into that spring device, and we'll squeeze the bag so that the fresh frozen plasma is sprayed into cell like that. Okay? So you'll have one bag with the platelet concentrate and then the other bag with the plasma, which will be frozen. Okay, so fresh frozen plasma can be stored for a year at -18. Red cells can be stored for as long as 42 days at refrigerated temperature, 4 degrees centigrade. And platelets are stored at room temperature, about 24 degrees centigrade. And it must be agitated gently. And it must be in a gas permeable bag, because the oxygen has to be able to get in. Okay, we don't want the platelets to start clumping. That's why it's gently agitated. And platelets, because it's not going to last long, it's 5 days maximum. So why do we separate the different blood components? Well, because you have different purposes for them. You know, red cells are used to carry oxygen. So if a patient requires more red cells, we want to just give them red cells. Fresh frozen plasma has clotting factors. So those patients that require clotting factors will get just the plasma. And platelets are useful for primary clotting. So we don't want patients that are in chemotherapy to get their platelet counts go so low that they might have brain hemorrhage. Okay, so each unit has its specific purpose. Okay, look at these. And which one do you think is the number one reason that we would transfuse? Is it accidents, cancer, gastric surgery, bleeding ulcer, pneumonitis, open heart surgery? How many think accidents? How many think cancer? Gastric surgery? Open heart surgery? How many don't know? How many people don't have arms?
Well, guys,
Where's my
I mean,
legs?
it's just
Yeah.
4 choices.
Okay, well, number one reason is cancer. Think about what sort of treatments are for cancer patients. They get radiation, they get chemo, right? What do those treatments do to the patient's cells? Nasty chemicals, nasty radiation. When you get irradiated, you're trying to inhibit the cancer cells from replicating, right? There's collateral damage when you give chemotherapy and radiation therapy. Normal cells will start to be affected too. So if your bone marrow is not able to produce enough red cells or platelets, these patients need transfusions. So the number one reason we're transfusing is because of cancer patients. They require a lot of support during their chemo and radiation therapy because their cells are being inhibited. And number one type of movement is probably platelets. And the next order is bleeding ulcers, disease of the GI tract, and neonates. So neonates, because they're really, really tiny, some weigh less than 2 pounds. Anytime we have to do a blood test on it, we have to record on the bassinet how much volume was removed. And if babies are really fragile and we have to test them frequently, that adds up. So that's called a nosocomial anemia. When we're the people that's causing the anemia, the baby's not anemic because it's losing blood on its own. The baby's losing blood because of all the testing. So when that volume of blood that's recorded for blood testing adds up to about 10% of their total weight, we transfuse. And there's a lot of neonates. If you go to visit Kupilani, there's a lot of neonates that get frequently tested that need transfusions almost twice a week. So, yeah, they comprise a lot.